All right, so today we are going to talk about protein synthesis or how we make proteins. So remember, proteins are one of our macromolecules and they're made up of amino acids. So the monomer of a protein is amino acids. So today we're going to learn how the DNA is used to link those amino acids together to make a protein. All right, so DNA has the instructions for all of an organism's proteins. So you are made up of proteins. That's what makes you up. That's what expresses your traits. That's what makes your body function on a daily basis. But DNA is stuck in the nucleus. DNA can't ever leave the nucleus. It's too big and it's too valuable. It's just like that recipe that's stuck in the recipe book. We can't take the recipe out of the recipe book. It's stuck. So we're going to have some way to get that recipe to the kitchen or get that recipe to a friend so the friend can make chocolate chip cookies or whatever they want to make. So the idea or the central dogma around what we're going to learn today is that DNA is changed into RNA, and then that RNA is used to make proteins. So on your notes on the very top, you're going to write what's highlighted in green. So DNA makes RNA, and RNA makes proteins. And it explains very simply how life is determined through all of DNA. So each section of DNA that codes for a protein is called a gene. The gene is read and the message is used to make a protein. And then proteins determine your traits, like your eye color, your hair color. Do you have dimples? Do you have freckles? All of that is determined by your genes. So protein synthesis, or the definition of what it is. Synthesis means to make. So protein synthesis means to make proteins. And there's two steps in protein synthesis. The first step is transcription. And in transcription, we're going to transcribe the DNA into RNA. A scribe is somebody who writes. So in transcription, we're going to copy the DNA into RNA. And in translation, the ribosomes are going to use mRNA to make a protein or to put everything together. So transcription is going to take place inside the nucleus. We have to copy that original recipe. We have to copy the DNA, and the DNA is too big to leave the nucleus. So we're going to have to copy it down. That original recipe that we have in our recipe book, we're going to have to write it down so we can bring it to a friend or make a copy of it to bring to a friend. We don't want to harm the original. So the players involved, what do we have here? We're going to have DNA, and then we're going to need mRNA. mRNA is our messenger RNA. It's going to carry the message from the DNA in the nucleus to the ribosome. So the mRNA would be Miss Neal. Miss Neal is going to copy the recipe from the recipe book and bring it to you in the kitchen. So what happens exactly? DNA is going to unwind. So the DNA is going to untwist, and it's going to make this shape here. This is called a replication bubble. The RNA polymerase comes along, and it uses DNA as a template, and it makes a copy of it. And once a copy is made, that copy can leave the nucleus now, and it can go to the ribosome. Remember, RNA is only one strand, so it's nice and skinny, so it can fit out of the pore in the nucleus and make it to the ribosome. So DNA unwinds, the DNA is copied into RNA, and the RNA leaves the nucleus. So here we have our strand of DNA at the top. And we're going to match it up. Remember, RNA does not have thymine. RNA has uracil. So A matches with U, C goes to G, T, trees have apples, Cars have garages. Apples have trees usually, but we're going to RNA, so apples are underground. And then trees have apples. And your DNA reads this in sections of 3 or in chunks of 3, just like when you memorize a phone number. Your area code is 601 or 901. You remember it in chunks of 3 because it's easier that way. DNA does the same thing. So G goes with C. Cars have garages. Garages have cars. Apples are underground. Trees have apples. And cars are in a garage. Now that our mRNA is made, it's going to come out of the nucleus because it's skinny, it's only half the size, and it's going to travel to the ribosome. So our ultimate goal today is to make a protein. But right now we only have an mRNA message. So that message needs to be translated. So translation is the next step. Our body can't do anything with just a bunch of letters. We have to translate the code. So first of all, we're going to transcribe it or write it down. Then we're going to translate the code. In cooking, that's putting all of your ingredients into a pot. In DNA and protein synthesis, it's a little bit different. So those sets of 3, like your area code, are called a codon. And a codon is a sequence of 3 nucleotides that code for a specific amino acid. So codons are like words, and you have to translate the words in order to get the right message. So the first codon is UAC. On the state test, if you have seen a codon wheel before, those codon wheels aren't usually on the state test. So we're going to use a codon chart. So the first letter is a U. So you're going to find a U in the first column. It's right here. The second letter is an A. So you're going to find an A across the top. And you're going to follow those across until you get to the box where they intersect. Now we know where UAC is. It's right here. And its amino acid is tyrosine, or TYR. So let's look at the next one. We have GCU. So we're going to find G in the first column. We're going to find C in the second column. We're going to go across and down. So GCU is going to be somewhere in this box. It's right here at the top. So it codes for the amino acid ALA, or alanine. You have these codon charts on the back of all of the clipboards in the classroom. So you always have access to a codon chart. So what are the 3 different types of mRNAs that we're going to talk about today? First off, we've already talked about messenger RNA. And messenger RNA carries the message as codons from the nucleus to the ribosome. Ribosomal RNA is the RNA that makes up the ribosome. And tRNA is our transfer RNA. And it matches what's called an anticodon to the mRNA codon, and it brings the correct amino acid. So let's look at how that step works a little bit more in detail. So translation, or that codon chart, reading those codons, takes place at the ribosome. And we need the messenger RNA, or the message that was brought from the nucleus. We need the ribosome to read the message. And we need the tRNA. He's like our sous chef. He's going to bring us the ingredients to put together to make our cake. So the tRNA is going to bring us the amino acids we need to put together to make a protein. So what happens? The mRNA has left the nucleus and it's going to find a ribosome floating around in the cytoplasm and it's going to bind to it. The codons on the mRNA are going to be red. The tRNA is going to bring the right amino acid that matches the codon. Remember, we use that codon chart. We use the mRNA on the codon chart to read what amino acid the tRNA needs to bring. Does it need to bring sugar? Does it need to bring salt? Does it need to bring flour? Does it need to bring licorice? We need to know. So the tRNA is going to grab that amino acid or grab that ingredient and bring it back to the ribosome. The amino acids then link together to form this word that my head is blocking is protein. So here's our ribosome. Our mRNA has arrived at our ribosome. Our first codon is the letter AUG. So AUG, if you look at your codon chart, codes for methionine. So the tRNA with methionine is going to come over and it's going to match up. These three letters are called the anticodon. They should match just like a DNA pair. And it lets this tRNA know it's in the right spot. So this tRNA will come, it's going to drop off its methionine. We're going to read our next three letters, which are UAC. UAC, if you look on the codon chart, codes for tyrosine. We're going to make sure the anticodon matches, and when it does, this tyrosine is going to bind. Those two amino acids are going to link together. We're going to go on to the next codon, which is GCU, and it codes for alanine. So a little bit of practice here at the bottom. If you want to try, we have TAC. So trees have apples. Apples are underground and cars have garages. So we'll use our codon chart and that amino acid this doesn't go there. So it translates the strand first. And then it matches the proper amino acids. You're going to do this same thing on your practice today in class.